Question number 3. Question number 3 show us the cripple allele in the chicken. Yeah. So this cripple allele, let's see here, cripple allele is a dominant gene. Yeah, it's a dominant gene. So that will means that all the cripple chicken, it will be the dominant trait, and the normal chicken will be the recessive trait. Yeah, recessive trait. Okay, so now, first thing first, it looks like a normal Mendel's model, right? Because we are not sure whether there's any relationship in between these two dominant and recessive allele or not. Okay, so in order to further find out, since they tell us that when two creeper chicken are crossed, yeah, two creeper chicken, yeah, they are crossed, and this is the number of the offspring that's being produced. They are producing 46 creeper chicken and 21 normal chicken. So based on this, whenever they give us the number of the offspring, we can use the number of offspring to find the ratio first. Because normally, the ratio of the offspring can tell us something. Yeah, they can tell us some relationship. Okay, so let's see. In order to get the ratio, yeah, so we will use the 46, yeah, divide by the smallest number will be 21. Okay, so 46 divide by 21, so it would be what? It would be 2.19. Yeah, the ratio is 2.19. Okay, so 21 divided by 21, it will be 1. Okay, so that is the ratio. Yeah, so the ratio is around 2 to 1. Yes, around 2 to 1. So if it is 2 to 1, the ratio is weird. Why? Because normally, normal Mendel's model, it would be 3 to 1 ratio, right? So this one is 2 to 1. So if it's 2 to 1, then I think there's only um, one type of inheritance that can link, that can have these 2 to 1 ratio, which is the little allele. Yeah, which is the little allele. So that's why for these questions, it is actually related to the deviation from the Mendel model. The deviation from the Mendel's model. Yeah. So it's related to what? It's related to the little allele. At the little allele. Right. So for little allele, how we should solve this question? It is still using the type 1 method. It is still using the type 1 method. Find the genotype of the parent. Then after that, find the possible gamete that can be produced. And then lastly, do the cross. It's still the same thing. Okay, so let's see the questions. So first question is asking, what is state the condition that caused this phenotypic ratio? So the one that caused the 2 to 1 ratio, I think that's only one. Yeah, so that's why that's the answer for A. Yeah, state the condition. The condition is what? The condition is the little allele. Okay, so next we look at B. So for B, explain how this phenotypic ratio is obtained. So how 2 to 1 ratio is obtained. Yep, so first thing first, same thing. We find the parent first. Yeah, you find the parents first. Okay, so for the parent, if you look at the questions, right? If you look at the questions, it is 2 crippled chicken, yeah? And that means that the parent are both having the dominant trait. Yeah, both also crippled chicken. All right. And they are able to produce normal chicken. Okay, 2 crippled chicken able to produce a normal chicken. So that actually gives you a hint that the parent must be both heterozygous. The parent must be both heterozygous. Okay, why? Because if let's say any of them, yeah, if let's say if you look at here, I can show you here. If let's say any of the parent is homozygous dominant and you cross with any heterozygous also, it won't work. It won't produce any normal chicken at all. Okay, so let's see. So this parent is going to produce only one type of gamete, which is big C. This parent is going to produce two types of gamete, which is one big C and one small c. Okay, so if you do the cross, you can see that all the offspring produced will be crippled chicken. None of them will be the normal chicken. Okay, so if you look at the ratio here, yeah, there are 21 normal chicken. So if any of the parent is homozygous dominant, that is impossible. It's impossible to get the normal chicken here. So that's why, yep, we can now confirm that it is impossible. Yeah, so the only possible genotype of the parent must be both heterozygous. Yeah, must be both heterozygous. So that's why this one is out, confirm out. So we no need to write that. Okay, so once we confirm the parent, okay, then next is you have to figure out what are the gamete that can be produced. So we'll do the Punnett square. Okay, so first parent is a heterozygous, so it is going to produce two types of the gamete, the big C and the small c. So second parent is also the same. Second parent also produce two types of the gamete, a big C and a small c. Okay, so let's do the cross. So it will be a big C, big C, small c. Okay, so after we get this, now we need to explain because they asked us, right? Explain how the phenotypic ratio is obtained. All right, so by showing this, so obviously what? Obviously, in order to get the 2-to-1 ratio, right? In order to get the 2-to-1 ratio, the 2 creeper chicken to 1 normal chicken. So from the Punnett square, you can see the 1 normal chicken is here. The normal chicken. Yeah, we have found the normal chicken. Okay, so how about the creeper chicken? The 2. The 2 creeper chicken must be the heterozygous one. Yeah, it must be the heterozygous one. Okay, so it is the 2-to-1 ratio. So what is missing here? The homozygous dominant C is missing. Homozygous dominant C is supposed to be creeper chicken, but obviously it is not born here. It's not born. All right, so that means that they has been dead as embryo. Right, all of these individual or all of these chicken with the homozygous dominant genotype, they are dead. They die, right? As embryo, they die before they get born. All right. So that's why, yep, this is how we explain that. Obviously, the creeper chicken here, the gene here, right? That is the little allele. Okay, so obviously it need two dominant C. It need two creeper gene, yeah, in order to cause death. Yep, so that's why this is what we can mention. The first point, we can say that the creeper gene, okay, the creeper gene, yeah, this is the first point that we can state. The creeper gene, the big C, is a recessive little allele. Okay, because it need two copies, then only then it will cause death. It need two copies, then only it will cause death. Yeah, if one copy, it will form creeper chicken. The creeper chicken won't be dead. All right, so that's why. Yep, that's the first thing that we can conclude. So second thing that we can conclude is, of course, the homozygous dominant C. Yeah, the homozygous dominant. Yeah, will be dead. It's dead as embryo. This is the second thing that we can confirm. Okay, so that's why these C, it will not contribute to the ratio in the offspring. You won't see them in the ratio of the offspring. Yeah, that's the reason. Okay, so what are the third thing that we can say? It would be, right, the third point, all the creeper chicken, right, it must be heterozygous. All the creeper chicken must be heterozygous. Okay, so with that, yeah, that's why we can get the ratio. Sorry. That's why we can get the ratio of 2 creeper chicken to 1 normal chicken. That's the reason of this ratio. Okay, so with that, yes, that is all for this question number 3.